Hello, everybody. My name is Véronique Chotteau. I'm working at KTH. This is the University for the Education of Engineers. We're located in Sweden and Stockholm. And I would go through some of the activities that we're doing, and then I would like to share with you our latest experiences with developing and understanding perfusion processes. So my group has been working with intensification in perfusion processes since quite some years now. So here you have kind of a representation of a continuous process in a bioreactor. And I'm currently director of at BioPro, which is a Center for Advanced Bioproduction by Continuous Bioprocessing, where we concentrate our efforts on continuous processes for the production of different modalities, biopharmaceuticals, but also viral vectors and cells for cell therapy. We're looking both at upstream and downstream, and also integrated these two. And around this activity, my group is quite active, but I also belong to other projects or other initiatives where we kind of complete our interest for high cell density perfusion with monitoring tools, with mathematical modeling. And these last years, we have been focusing more and more also on the production of viral vector. So from this intensification aspects, now we're kind of navigating through different projects. But today I will also share with you data that we have had in different constellations where the Wallenberg Center for Protein Research was also active. And I'm also coordinator of a EU project, which is iConsensus, where we're looking at developing new tools for real-time monitoring of a process, which should be outlined, where we also look into modeling, mathematical modeling, and even development of microbioreactors. And here you have a flavor of the different activities and the partners in this project. And actually, Irvine Scientific is also not really a partner, but sponsoring also our efforts in this group, especially at KTH in my group. So now looking into what we have been doing since many years into perfusion. So in the top corner, you see the concentration, high concentration densities that we obtain in wave bioreactors. That was actually almost 10 years ago now. But after that, we have been concentrated our efforts more on stirred-tank bioreactors. So now we're speaking about high cell density perfusion process, the way you kind of envision it with a bioreactor, continuous medium renewal, a system for cell separation, where you have the concentrated cells which are maintained in your bioreactor, and you have cell-free supernatant that you can dispose or take forward to the downstream processing. So we started in 2010-11 with obtaining cell densities which were above 200 million cells per milliliter. And we were the first group to present these type of data. And at that time, we worked with wave bioreactors. And in this complicated graph that you might have seen before, actually, what is nice to see is that we developed a system where the viability was nicely maintained at a quite high level. At the same time, we were kind of able to push the cell density, which are represented by these different curves, to more than 200 million cells per milliliter. What we also did was to maintain the cell density at around 100 and then around 100 million cells per milliliter continuously by just bleeding the cells. What we have done at this time and what we still do today is to apply cell-specific perfusion rate, which is a way to provide the nutrients to the cells at a kind of constant way, and also to discard the byproducts. What was also nice to do at that time, and which is still something that we apply a lot, was to, when we were at lower cell density, to maintain the system around 20 or 30 million cells per milliliter and explore different parameters of the system. This is not obvious from here, but this is basically what we've been doing at that time and what we are still doing today when we do process development, to learn what is happening. And then when we have decided what we want to do, then we can push up the cell density and get application of our results to higher cell densities. So here you have on the top, or on the right hand side, you have a flavor of the results that we have obtained with different systems, different cell lines, of CHO cell lines, and more recently of HEC293 cell lines, for the production of antibodies, different types of antibodies, but also non-antibody glycoprotein. What you can see here is a presentation of the cell density as a function of time here, where you see that we have obtained repeatedly really high cell densities, and also kind of repeatedly we have been kind of working around 20 or 30 million cells per milliliter before we kind of increase the cell density in order to just repeat our development at the higher cell density. From this picture, what you can also see is kind of an interpretation of how the field is viewing high cell density perfusion today. So what you could say is around 20 million cells per milliliter is the state of the art for bioreactors today, or maybe already yesterday, because the new trend are actually now to increase the cell densities. And this is already applied in a really efficient way at N-1, but people are also thinking about that for production. And the vision is, depending on who you're speaking with, it can be cell densities which are around 100 or higher cell densities. So these data and others, you can find them in publications that we have been working with and that we have published. And here you can see also in the down corner different systems that we worked with, the wave bioreactor, the stir tank bioreactor with ATF, and actually also with TFF, but also cell tank, which is a kind of different system. And I refer to publication for that system. So what is of course nice when it comes to high cell density perfusion is the benefit that you get, the ability to produce in a much smaller footprint, much larger amount of product. And this is illustrated here in this picture, where you can see that in this accumulated product from a perfusion, which was actually performed at 2 cell densities, which is the reason why you have different angles here in this accumulation curve, you can see that in comparison with the production that you get from a fat batch during something like 2 weeks, you accumulate much more product in terms of volumetric productivity. So you can produce in 1 day the same amount as you have in a whole process of fat batch. And what illustrates that well is also the volumetric productivity, the amount of product per working volume per day that you have as a function of the viable cell density. So what you're looking for is something which is increasing. So the more you increase the cell density, the more you have, the larger is your volumetric productivity. So by obtaining this pattern, you obtain a system which is really efficient, which allows you to really produce a lot of material in a really small footprint. What we looked also at the time we achieved really high cell densities was to look into what is the effect of this high cell density. And this is kind of more from a mechanical point of view. So if you think about the theoretical distance between the cells when they are in culture, so just considering them as perfect spheres, what you can see is that when you plot this distance as a function of the cell density, when the cell density is not really high, the distance is really large, but as the cell density is increasing, this distance is rapidly decreasing, and it comes to a point where the cells, where this is kind of reaching zero. So that means that the cells are against each other. That means also that they are against the wall of the filtration system. And if you enlarge what is happening here, you can also see that in detail that it depends a lot of the diameter of the cells. So if the cells have, for instance, a diameter of 16 micrometer, with this theoretical calculation, they come to this point where they are against each other at the cell density of 250 million cells per milliliter. And of course, you can imagine that in this case, the system will have a totally different behavior, because you will not have this kind of fluidity in the movement as the cells can have when they are more freely in the culture. And it also translates into increased viscosity and also increased fraction of the cells occupying volume in the cell suspension. So to the right here, you can see a representation of the volume fraction of the cells as a function of the cell density. So this increased linearly, and what you can see is that around 200 million cells per milliliter, when the cell density, when the cell diameter is about 17 micrometer, you have suspension where the cells occupy 50% of the fraction of the volume. So this is really thick. To compare with something which is physiological, this is actually what is happening in the blood. So the blood has about the same amount of red cells in comparison with the volume occupied by the red cell is the same in comparison with this system, is about 50%. And to the left, what you can also see, without going too much into detail in this kind of more complicated picture, but what is interesting is to see that the viscosity is increasing quadratically with the cell density. And it follows this relationship, which is the same as the relationship that you have for all other colloidal systems. And the thing is that it increases quadratically. So when you have a system which is at rather low cell density, you don't really see so much of an effect of the cells. But when the cell density increases, this is something which is much more dramatic. So this was kind of the mechanical part. We looked also at how this system behave when we're pushing the cell density, but still applying a cell-specific perfusion rate. And here, this is my previous graph, but represented completely differently. So with the cell density as a function of time, and the red dots represent different samples, which were taken to look both at the metabolomics and the proteomics present in the extracellular part of the sample. And this is compared here with fat batch, which were performed with the cells and the same medium. And here you have an enlargement of this fat batch, kind of really typical fat batch. And here you have a representation of MS for the metabolome and for the proteome. And what you can see is that if you look at the principal components of the system, for the fat batch, the system is kind of evolving a lot. There's a lot of dynamics, although the cell density is not changing so much compared to the perfusion. In the perfusion, you have cell densities which are stretched from 5 to 200 million cells per milliliter, and the run is much longer. But you can see that these data cluster much better compared to the fat batch, where you have a lot more dynamics, which is occurring here. And the same is true for the proteomics. So this is the representation of about the same thing. And here you can also see how the proteome is kind of consistent with itself at different days of perfusion. what is interesting, though, is that when you increase the cell density to really high levels, you begin to see really events which are different between low and high cell density. And what we observed was an increase of the cytoskeleton and the stress fiber proteins, and also a modification of the glutathione metabolism. And when you look at what is happening when there's stress fiber assembly, which is, for instance, characterized by an increase of the actinin, it is known that this is important for the cell morphogenesis, and it means that also the cells are reacting to external stress, to mechanical forces. This is also a function which is important for the contractility of some cells. So that means, or you can represent that the cells are actually reacting at this extra shear stress, and they are kind of building the stress fiber assembly and becoming kind of a little bit more robust and also more contracted, which can explain why we and others actually have observed that at high cell densities, the cells can actually become smaller, so have a smaller diameter. What was also obvious in this study is that the glutathione metabolism became different when the cell density was pushed to high levels. Switching gears a little bit, with time, what we realized was that we didn't want to work with systems with 4 or even 1 liter as a working volume, but we wanted to have a system which was really scaling down that. And this is kind of a little bit busy slide showing the different aspects that you should think about when you develop perfusion processes. And to the right, you have kind of the tools which are accessible for process development. And what is really missing here is small bioreactors, which can sustain high cell densities, but which would be of small volumes. So we looked at what was possible, and we were targeting 200 milliliter. And we did the development of the system. We actually did that kind of in parallel with the Amber. So this was kind of completely separate from the efforts which have been put to develop the Amber perfusion. And this is a flavor of the data that we got in the first hand with looking at the system for CHO cells. So here you have a representation of the cell density and the viability as a function of time in a system, which was the small bioreactor, 200 milliliter in the task box system and in the multiforce system, which is actually a little bit higher in volume to 400 milliliter. And both systems were connected to hollow fiber, which was coupled as a TFF system. So what you can see is that we could obtain a system where we could push the cell densities. And here you can see different levels. These are levels that we have stopped in order to study the system, to be sure of what we have before we increase the cell density to higher levels. So in both systems, we could obtain cell densities up to 100 million cells per milliliter, and we could maintain actually the viability at reasonable levels. However, what happened is that, up to 100 million cells per milliliter. But a purpose was also to apply the system to the HEC293 cells. But when we wanted when we start doing that, then the foam excess that we actually have in this system and the high shear stress by the system was not tolerated by the HEC293, which are more sensitive. Then we started to kind of dig into understanding how we could improve this system. And I think that the findings that we have gathered with this system are actually also important for the field, even for larger systems. So there were kind of 2 types of problems that we were faced with. One were more from a mechanical point of view, that the cell separation device generated a high shear stress. We needed to get rid of the excess foam. So this kind of more hydrodynamic optimization. And on another hand, what we also saw is that in this system, like in other systems, we have had problem of too much lactate accumulation. And actually, one of the problem has come from the fact that potentially the delivery of the glucose is not really in harmony with what the rest of the medium is bringing as nutrients. So then we have developed a strategy for glucose delivery, and I will not speak about that now. It has actually been published now. So we apply a targeted feeding called TAFE, and this is a way for us to ensure that the glucose is delivered just to corresponding to the cell demand, and also that the lactate accumulation is not too high. I will not have time to speak either about the optimization of the oxygenation, which has been performed through optimization of the impeller system, but I would like to concentrate a little bit more on the cell separation device. So you might be familiar with the graph which is represented on the right, which is coming from the work of Chalmers group, where you recognize different types of shear which occur in a bioreactor, like the agitation, the mechanical damage in a flow-through pipe, and the bubble rupture. And on top, you see how different cells can tolerate this different level of energy dissipation rate. And to the left, you recognize the ATF and the TFF, alternating tangential flow filtration and the tangential flow filtration. Both are equipped with a hollow fiber system, but the movement in these is actually different, as you know. And in both cases, the passage through the hollow fibers relates to flow through pipe mechanical damage. So we looked a little bit closer at what it meant in terms of shear stress and the shear rate. So if you think about the TFF, the system has a flow circulation in the hollow fiber, which is constant. While in the ATF, the cell growth is going in and out. So here you have a representation of the velocity as a function of time, so during one cycle. So on the red bar, or the red line on top, is for the TFF. So this represents that the velocity is constant. While for the ATF, the fact that the cell suspension go in and out in the filter provokes actually a sinusoidal behavior. And if you take the average of this velocity curve, you can actually see that the velocity and the shear stress, as well as the shear rate, are actually smaller in the ATF compared to the TFF, according to this relationship. So the shear stress that you have in the hollow fiber of an ATF is around 0.64 what the shear rate is in the corresponding TFF. So, and you can also see that from a more experimental point of view, and this is what we have done. So we have studied the system where we had bioreactors which were equipped like in perfusion, but the culture were short, they were only batch cultures, and during which we did an exercise to look at different flow velocity through the cell separation device, and also both in ATF and in TFF. And here you have a flavor of these results that you can also look up in our recent publication. So this is a representation of the growth rate as a function of different flow rates for the ATF in blue and the TFF in red. And what you can see is that with the TFF, when the flow rate is increasing, the growth rate decreases from a certain point. And you have also a decrease, which is observed with the ATF, but it's actually a little bit later. And this actually relates really well to the relationship, which can be deducted just from the theoretical approach that I described just before. So from these data, both from a theoretical point of view and from the observation of the growth rate, which is different in these systems, you can actually see that the effect of the ATF is milder compared to the effect of the TFF for the cells. Then we took samples that were generated during this study, and we looked at how it looked for the gene expression. And you can see here that in regime where you have low shear stress or high shear stress or no shear stress, you have kind of different genes which are expressed or not expressed. So you have really differences which are coming. Now, I will not have time to go so much into detail, but when you have high shear stress, you have a series of genes which are increased, which relate to the fact that the metabolism becomes less efficient. You have an increase of the unfolded protein response, which is a way for the cells in the first place to kind of decrease the protein translation in order to keep up with the production. But at the end of the day, it also provokes the apoptosis. You also have an increase of the oxidative stress and other manifestations of stress for the cells, like increase of the inflammation. And here is also a representation how apoptosis is taking place when, in case of high shear stress, it goes through 3 different mechanisms: cytoskeleton reorganization, ER stress pathway, and also extrinsic signal inducing apoptosis. So this is probably not a surprise that apoptosis is triggered with high shear stress. What was interesting, though, was to observe that moderate shear stress is actually beneficial to the cells. So this is illustrated in these curves here, where for the TFF and for the ATF, when you look at the relative EPO expression, the mRNA expression as a function of the shear rate. So when you would have one, which would be the level of no shear, the control level, you can see that when the shear rate is low, you actually have a value which is much higher than 1. So you have increased the EPO mRNA expression. And of course, when the shear stress is increasing, this is actually decreasing. What you can also see is that it provides increase of the expression of genes which are related to the cell division, the cell proliferation, the mitotic centrosome, and also some protection against apoptosis. What we also could confirm in this study is that we identified a certain threshold above which the growth rate and the metabolism would decrease, or would have performances which would decrease, and these were kind of well in agreement with the representation or the work of Chalmers, looking at different range of hydrodynamic conditions to provoking cell destruction through agitation, so through pipe or bubble rupture. So when we had optimized this system, we of course could use it. And here you have a flavor of the work that we have done. Here are data for HEC293, but bear with me that if we can make the cells, the HEC293, nicely surviving and producing well in these small bioreactors, that means also that we have a system which is favorable for CHO cells. So here you have a representation of 2 runs which have been performed with HEC293 cells producing EPO, where you can see here in the first run that the cells have been increased to a cell density around 20 million cells per milliliter, maintained by cell bleed, and kind of looking at different conditions. And then the cell density was increased by different steps. While in the second run, we knew from the first run that this was a system which was well in place. So we increased the cell density at once to a higher level. And then we could even push this cell density to a much higher level. The viability was nicely maintained at really high, much more than 90%. And here you have also a representation of the CSPR, the cell-specific perfusion rate, which was studied also in this system, starting around 50 picoliter per cell per day. It was actually decreased during the study of the second run. And what we could see is that the cells had still a nice behavior. Here we actually decreased the temperature, which was the reason why it decreased. But otherwise, the system could be well restarted or regrown later in the culture. So one thing which was also important was to kind of being able to show that the same way that we had a volumetric productivity, which was increasing with the cell density with CHO cells, we could do the same with HEC293 cells. And this is illustrated here. For EPO, you have a representation here of the units and here of the milligram per liter per day as a function of the cell density. And here you can also see that you have a nice increase of the volumetric productivity as a function of time. So coming now to summary and perspective. So we started, and I kind of was happy to bring our old data here, where we reached more than 200 million cells per milliliter at that time in wave bioreactor. And since then, we have been doing a lot of work, mostly in stir tank bioreactor. And lately, we developed mini bioreactor of about 200 milliliter. Which is now a really nice tool, really popular in my team in order to perform experiments both in CHO cells and in HEC293 cells. In this system, we can maintain the cell density at 100 million cells per milliliter, or even a little bit higher. And we have a behavior which is quite comparable to our bench bioreactor of 1 liter in terms of metabolism or productivity. And this is also, it's also nice to see that even with the HEC293 cells, we're able to maintain the productivity in a quite efficient way. We also digged a little bit more into what perfusion really meant for the cells in terms of what is the effect when the cell density is increased to really high level. So what we could see is that there's an effect of on the really high cell density on the cytoskeleton. And this is also a nice explanation of why we and others have observed that the cells become smaller when they are at really high cell density. We have also seen some effect on the glutathione metabolism. We have shown that the ATF in itself, just by its design, generates actually less shear stress than the TFF. We have also supported that with experimental data. And what was also interesting to see is that when we look at the gene expression, we can see, of course, that high shear stress is not beneficial for the cells, which is probably not so big surprise. But when it comes to limited shear stress, we can see that actually it can have a beneficial effect, both on the recombinant EPO mRNA expression and also on the cell division. And with that, I would like to thank you for your attention. And I will also like to thank my team, represented here to the right, and also collaborators from the group of Professor Johan Rockberg and Professor Adil Madinoglu. And I would like to thank also the sponsors. Thank you.
All right, thank you very much, Veronique. And so we'd like to continue the presentation now with just kind of a wrap up here. And this will be, this is Tom Fletcher from Irvine Scientific. And let me see if I can, yeah, can you see my screen, Veronique? I just want to make sure. Yep. So, yep, so we're very pleased to have this presentation today on perfusion because it's such an important subject for us at FUJIFILM Irvine Scientific. So we're very active developing improvements to media explicitly for use in perfusion. And so I'd like to just give you know, some overview, some flavor of that very briefly today. And first of all, just for those of you who may not be familiar with our history, I just wanted to point out that we are one of the oldest media suppliers in the industry. We've been in business now for over 50 years. And also we have a fairly rich history in terms of developing serum-free media. And we're actually the very first company to offer commercial serum-free media for CHO, for hybridoma, and also for HEK293. So that goes back, you know, quite some years, but we've been very busy in those areas until now, finding ways to improve the media. And over this long history, you know, of course, now we're focused mainly on chemically defined media and looking for ways to always deliver the best media possible for large scale bioproduction. So when we think about perfusion culture, and as we read the literature, we find that there's many different ways that perfusion is used. And Veronique mentioned several of them, you know, the N-1, the hybrid fed batch, intensified fed batch. And so this slide is a bit busy. It's just kind of a summary of some of the different ways that perfusion technologies are being used. And when we thought about it, what we really recognized was that the one area that seems to need help with media, and of course we hear this from our customers, we really need better media for steady state perfusion. And so that would be continuous culture. And so this is where I think the medium is, you know, the demands for media, the requirements are quite different. So you might be able to use fed batch media for just about everything on this slide, except the steady state is where I think we really need to find ways to improve the composition of the media, especially with the concern for how much volume of media is required at high density. So, you know, as you consider perfusion for a large scale process, you quickly realize that the quantity or the volume of media is a concern, and the cost of that media. So we really wanted to find ways to reduce the amount of media required, and of course, support the performance of high density, steady state perfusion processes. And so again, since we've learned a lot about nutritional requirements over the years of developing serum-free media, we also wanted to distinguish between simply screening media or blending and screening media. This would be what I consider a random approach for identifying very quickly, you know, the best medium. And so this is a common practice. We also begin with screening every media development project. But in the middle of the slide, I've highlighted more rational methods. And this would be really considering spent media analysis, considering, you know, the cell-specific rates of consumption, production, and really trying to understand where are the best opportunities to improve the nutrient composition of the media. And so using these type of information, we're able to use a push to low approach. And so this is a common approach that, you know, Veronique mentioned the CSPR, the cell-specific perfusion rate. And so this is a good indicator of, you know, the goal that we have in mind, and that is to minimize the amount of media on a cell-specific basis required for any production process. And then, as you can see in the green bullet points here, just the inverse of a CSPR would be what's referred to as the medium depth, and that would be cells per milliliter per day. And so these kind of metrics are actually described very well in a paper by Konstantinov, and I'll be happy to provide that reference, but it is kind of a landmark paper on the subject of using a push-to-low approach to improve or to optimize a continuous perfusion process. The other subject that's at the bottom of my slide here is modeling. So again, it was touched on briefly by Véronique, but when we do media development, this becomes a very obvious problem that we need to address, and that is, how do you evaluate many different conditions, many different media, and it demands really some thought and some development of small scale models that allow you to have the throughput that would be meaningful to compare various media. And so we've used quite a few different combinations of spin tubes, microbioreactors, thinking also about cell retention and, how is the media actually exchanged. So just an important topic I want to mention. And then just briefly, I won't spend a lot of time here, but just describing what a push-to-low approach is. There's several ways that you can push down the CSPR. One of the most obvious ways is to reduce the exchange rate of the media in a steady state. And so that was exactly what some of our colleagues in Japan did. And I'm just showing one result from their media evaluation. And so this was just a single bioreactor where they used a push-to-low approach, basically reducing the media exchange rate over the course of a steady state culture and achieving a nice low exchange rate while maintaining a high productivity of 1.5 grams per liter. So again, quickly just calculating what they've achieved here, they did push down the media consumption down to 12 picoliters per cell per day, which again, you can just look at the inverse of that as a measure of the nutrient depth or the medium depth, which would be 83 million cells per milliliter per day. So some very useful metrics. And I just mentioned it for that reason, both in terms of technique and in terms of what are we measuring in terms, to identify performance when it comes to a continuous perfusion cell culture medium. So these are some of the things we're looking at. And so again, that was very brief. I apologize. I'll be happy to tell you more if you'd like to follow up. But how can you benefit from some of the things we're learning here at Fujifilm? Well, we have media development services. We'd be happy to develop a medium for your process. We offer that as a complete optimization service. We also can support your own development through spent media analysis and what we call express media service. So that's a service where we can rapidly deliver samples for you to evaluate. They can be based on your own formulas, and it just allows you to spend your time on, the evaluations. We'll be happy to make the media to support your experiments. And the second thing on this slide are media survey panels. So this is actually one of the easiest ways for you to get to know our capabilities. We have panels of media that are ready to go. And again, you can simply screen these in your own process with your own cell line and understand, kind of a range of options and how well they perform for you. So these media panels are designed, explicitly to support particular applications. We have been focusing mostly on CHO, but we're quite excited about the potential for perfusion in HEK293 as well. And so we're working in both areas now, and it's an exciting time for us. So I went kind of quickly, and I apologize, but we will be happy to take questions either now or please follow up with us later. So I think we'll just end there and possibly take a few questions. So let's see. We do have a few questions here. Véronique, I see a question here that's addressed to you, and you talked about, some of the benefits of a moderate level of shear. And so the question is whether those effects extend also to product quality attributes, so critical quality attributes. Can you comment briefly on that?
Yeah, it certainly does, actually, Tom. This is a really relevant question. So we have not done this kind of work, but this is work that people have done. So you can see there are really good reviews on that, kind of showing that mechanical shear is actually potentially damaging the product. And it's actually not only during the purification, but also during the culture. So this is always a relevant question to really think about, definitely.
Okay, and then I had my own question on the moderate shear. I was just curious, how did you distinguish, the benefits from the potential effects on, if you're using a cell separation technique, perhaps the shear is related to some other, gas exchange rate or nutrient supply rate. How were you able to distinguish that the increase in shear wasn't simply improving something else like that?
Yeah, so I mean, the system that we took was a simplification, a model for the perfusion. Mm-hmm. So it was a bioreactor which was connected to a perfusion system, and with the perfusion, with the recirculation of the cell suspension in the cell separator like usual, but it was actually not really growing to really high levels. So it stayed kind of a batch process. And you know that batch processing is one of the processes which is most known by everybody. So this was the way that we could really maintain the system kind of under control. Low cell density, abundancy of a medium because it was just a short batch. So this was kind of the model that we used. So that was the way we did that. But this is certainly a really relevant question. I mean, we work both on how to find, how to optimize the medium and these kind of questions. So they are both important. That's really relevant.
Okay, I understand now. So it was a circumstance in which the gas exchange rate or the nutrients would not be limiting. It seemed that you were at low densities. Okay, and then let's see if I have another question here. Well, you talked about the, looking at some of the X-proteome and X-metabolome. And so, again, when we talk about shear, can you mention some ways that might affect glycosylation or some other aspect of product quality? I mean, you mentioned that it was damaging in the earlier question, but can you speak specifically about glycosylation?
I'm not sure that it can affect the glycosylation. I'm not specialist on glycosylation, although we have done quite some work on that recently, but more from mathematical modeling point of view. But I would be more thinking about the aggregation, because the aggregation, which is well known to spontaneously occurring when the product is purified, this is something which can also happen in your culture. So it will be really product specific. So it's really difficult to generalize, but I would be more afraid of aggregation.
Right. Okay. All right. Well, I think that's all we have time for. So again, I appreciate you, Véronique, being available for this. I hope it's been a benefit to all of those who've listened. And we appreciate the chance to share some of our work with you all. So thank you very much.
Thank you, Tom. Thank you for the opportunity.
All right.